contents of the cell, the nucleus, and how the cell uses DNA. The DNA, nucleolus, and nucleoplasm are all encased within the nuclear envelope. This is a lipid bilayer, just like the plasma membrane on the outside, that's studded with small pores to allow for small molecules, some very small proteins and RNA, to move through across the plasma membrane. Large molecules are generally excluded. Proteins and RNA sometimes, small molecules most of the time. Inside the nucleoplasm, most of the space of the nucleus, is where you find your regular chromosomal DNA. In the nucleolus, this tiny, tight little ball that you see in a lot of drawings, is where ribosomes and ribosomal RNA component parts are made. DNA within the nucleus is the operating manual for the whole body. Every cell has all the DNA your entire body needs. Your skin cells have skin DNA and kidney DNA and liver DNA and eyeball DNA. Your eyeballs has all those other kinds of DNA in it. Only certain parts of it that cell needs are going to be turned on, so to speak, at any given time. The DNA within your cell, that huge whole operating manual, isn't kept in just one big book, in one big tome. It's broken down into 23 parts. If any of you are old enough to know the Encyclopedia Britannica or have seen it on your parents' shelf or your grandparents' shelf, it's a huge body of information that's kept within that encyclopedia. But it's so much that to keep it in one giant book is wildly impractical. You'd have a hard time turning the pages or even lifting the thing. So they break it down into a bunch of smaller books. Your cell does the same thing with DNA. In people, we break that down into 23 parts from each parent. We have 23 copies from mom and 23 copies of complementary information from dad. You have about 6 feet of DNA packed into each and every nucleus of your cell. That DNA is double-stranded. So you've got DNA in that familiar double helix shape. Whoops. With the nucleotides, strung between them like a ladder. So the DNA is double-stranded and it's wound into the familiar double helix shape. So you have it sort of in this coiled sort of look. This double helix is then going to coil into a spiral. This is going to circle around proteins that look sort of like yo-yos, called histones. Those histones are going to coil up on themselves. And in this way, you can pack a lot of DNA into this very small space. 6 feet of DNA in the nucleus of every single cell in your body. It saves a ton of space, and it also prevents DNA from being damaged. If this is the outside of the DNA, you have a lot of your nuclear code packed into the middle here. You have a lot of it sitting inside each one of these little coils. That's a lot harder for the environment to affect. If it's not on the surface, it's hard to manipulate. A lot of the DNA is therefore kept away from the surface of a chromosome. A chromosome is each one of these 23 parts. We have 23 pairs, so 46 total chromosomes. When you have the double helix wound around those histones, those yo-yo looking histones, your DNA changes name and is now called chromatin. When the chromatin coils up upon itself again to make more of like a cluster, that's called a chromatid. This is only going to be found immediately before cell division. Typically, in a resting cell, your DNA is packaged in a form called chromatin. Everything's wound around histones, but they're not bunched up too much. During cell division, those chromatids can organize into the classic X shape that you're familiar with called a chromosome. Here's a picture of that. So you have the familiar double helix that gets twisted around itself until you form a tight spiral. Those get wrapped around histones, and this is the way your cell basically keeps it in your body. This is still a pretty tight packing. You can still have them sort of packaged back and forth next to each other to afford yourself a lot of protection and space saving. But only when they bundle up into very, very, very tight clusters immediately before cell division are you ever going to refer to a chromatid. Those, in turn, spiral up. Those spirals spiral back up. And eventually those spirals organize themselves into this familiar X-shaped chromosome. Oops, can't seem to erase this. I'll have to let it go for the moment. RNA is the working copy of genetic information that's specific to a given activity that you need to happen. So the analogy I like to use is that your DNA is the master manual for everything that has to happen in your whole town. It tells you where every building is, what every building is made of, how many windows every building is, how many shingles are on every roof, how many stones are in every sidewalk. That huge book lives in your basement, in the nucleus of the cell. If you need to go up onto your roof and replace a brick in your chimney, you don't want to drag that whole huge book all the way up to the roof to fix that one brick. Instead, it's much more efficient to just look through the book in your basement, find the instructions about replacing a brick on your chimney, copying that, and bringing that copy of the instructions up to do the work. DNA is analogous to that book in your basement that tells not just what's in your house, but in the whole town. DNA in your cell tells you not just what to do with that cell, but with your whole body. When you find a specific set of instructions to copy and bring someone somewhere else, that copy is what your RNA does, specifically your mRNA, your messenger RNA. RNA is a single strand of genetic information that contains all of the same nucleotides as DNA, except instead of T, you have a U for uracil. We went over this earlier with the macromolecules of life. mRNA is the message to go forth and do other work, to build other things, to do other work. It's like the copy you're bringing up onto the roof to replace the brick in your chimney. It's a set of instructions that goes to a machine, an organelle called a ribosome, to tell the ribosome what to make. rRNA is a special kind of RNA that's made in the nucleolus and makes up the structural component of ribosomes. Ribosomes are made up of proteins. These are the organelles in your cells that make proteins. They themselves are made up of their own proteins and a bunch of genetic information, a bunch of RNA. This special kind of RNA is called rRNA. tRNA is for transfer RNA. This is a special molecule that takes on a special funky puzzle piece kind of looking shape that has 3 nucleotides on one end of it. These transfer RNAs, along with their nucleotides, are going to get fed into ribosomes. When a ribosome reads a copy of mRNA, it's going to see what letter is here, find a tRNA with the opposite letter, and staple that down. As the mRNA moves through the ribosome, you're going to add another one. It's going to read a new letter, and it's going to add a new transfer RNA, and that's going to add a new transfer RNA, and so forth and so on until you have this primary sequence of a protein, the string of amino acids, the primary sequence of a protein. Here's a video about how DNA is packaged. It's not embedded in this slide anymore, unfortunately, but I do provide a link to it, both in this unit item on Blackboard, as well as in the master list at the bottom of this module page. This shows the double-stranded nature of DNA. It shows how DNA is organized into coils upon coils upon coils. It shows histones and its relationship to DNA. And it also shows a chromatid being wound up into a chromosome. Hopefully, after watching this video, if you still find yourself confused about how so much DNA is packed into the tight little space of the cell, hopefully this video will help clear it up. Now, we've talked about how you have DNA in the nucleus of your cell, but you only want to copy a little bit of it into RNA. The process of copying DNA into RNA is called transcription. RNA is written in a language of nucleotides. DNA is written in a language of nucleotides. Since you're copying the same information over in the same language, you're just rewriting it. The fancy term for rewriting something is to transcribe something. Make sure that you get this term exactly. The next step in the process has a term that's very similar to this. Getting them confused with one another is going to give you 0 credit on kind of everything that you do when you're asked about this stuff. So pay very close attention to the terminology here. What happens, and I'm going to breeze through this a little bit. I trust that you've gotten this in introductory biology before, so we're just going to hammer out the basics again. A molecule called RNA polymerase is going to come into the nucleus through one of the nuclear pores, find the section of DNA that it needs to be transcribed, and reads it. The DNA is going to be unzipped so that it takes on a regular sort of ladder-like appearance. The polymerase is going to come through and find a special sequence in the DNA called a promoter. The promoter tells the RNA polymerase, start transcription here. That's all a promoter does. It's a little signal that tells the RNA polymerase. Polymerase is an enzyme that makes stuff. RNA polymerase is an enzyme that makes RNA. It tells the RNA polymerase, start reading DNA here to make your RNA. That enzyme, the RNA polymerase, go through here and make more. I know they don't line up perfectly, but you get the point. The RNA polymerase, once it latches on, is going to whip down the DNA. Behind it, your DNA is going to coil back up into the familiar double helix shape that you're used to. The RNA polymerase is going to continue moving down until it sees a several letter sequence again called a terminator. At the terminator, the RNA polymerase pops off. In the meantime, while the RNA polymerase was zipping down the line and reading that DNA, it was creating behind it its own single strand of RNA. Where it reads one letter, it writes the opposite letter. If it reads a C here, it's going to write down a G in its RNA. If this is a T, it's going to write an A on its RNA. The final product is your original DNA double helix again, because it retwists after the RNA polymerase zips across it. And you'll have a single strand of RNA that includes the instructions to do a specific task in the cell. More specifically than that, to end up making a protein. The process of rewriting DNA into RNA, that short set of instructions for a specific task, is transcription. Here's another image of RNA polymerase doing its thing. You can see how in reality it's really a blob that kind of encompasses the whole double helix DNA strand. It latches on at a place called the promoter, which you'll find right about there, where you can see it makes its initial contact. And as the RNA polymerase moves across, it adds new nucleotides that are complementary to whatever it reads on the DNA strand that it's reading. If it reads an A, it's going to put down a U. Remember, it doesn't have thymine, it has uracil. If it reads a G, it's going to put down a C, guanine for cytosine. If it reads a cytosine, it's going to add a guanine. And if it reads a thymine, it's going to add an adenosine. Complementary base pairing applies to RNA transcription as well as DNA replication. It's important to note that ahead of RNA polymerase, because it's moving this way, DNA is wound up. Behind RNA polymerase, DNA also is wound up. The more time that DNA spends wound up, the less opportunity that there exists for environmental effects to damage any of those nucleotides. The more twisted up the DNA is, the better protected its contents are. Here's a video about DNA transcription into RNA. The cool thing about this video and the last one and the next one too is that these are happening in real time. So as fast as you see it happening on the screen is as fast as it's actually happening millions and millions of times over in every single cell in your body. It's really cool. It's so fast. But in this video, you're going to find you're going to see the RNA polymerase finding the promoter, unzipping the DNA after the promoter, zipping down the DNA and making an RNA strand behind it, and then when it gets to the terminator, popping off and releasing the RNA strand into the cell. The process of writing RNA, turning RNA into proteins being the things that are actually going and doing stuff in the cell, is translation. RNA is written in, oh, that's too dark. Let me try a different color. RNA is written in a nucleotide language, and proteins are written in an amino acid language. You're turning something from one language to another, therefore it is translation. DNA into RNA, just to hammer this home, RNA equals transcription. RNA into protein equals translation. So what you're going to have is an organelle called a ribosome. We've mentioned this before. A ribosome is the organelle in the cell whose sole responsibility is to read messenger RNA and translate the code in that messenger RNA to a string of amino acids. The first string of amino acids made by a ribosome is the primary structure of the amino acid. As it's ejected from the ribosome in this string, each amino acid, the R groups, are going to attract to one another or not, kinking itself up into either a zigzaggy kind of sheet or a coil. And that's the secondary structure that happens spontaneously. So the RNA, the mRNA, is fed through the ribosome like ticker tape. Each 3 nucleotides is referred to as a codon. These 3 nucleotides are read by the ribosome. The ribosome will then bring in the appropriate tRNA with the complementary letters to the 3 letters it reads here. The other side of the tRNA, which we didn't mention before, of course, is attached to an amino acid. Each 3-letter codon represents a specific amino acid. They're not interchangeable. TTA is always, if memory serves, that's aspartic acid. There's more than one codon for aspartic acid, but TTA doesn't ever mean anything other than aspartic acid. The ribosome will bring that amino acid in. The complementary nucleotides will bind, and a protein will, or an amino acid will be locked down. It'll shift the whole thing over one. It'll read the next codon. It'll bring in the complementary tRNA to what it reads on the mRNA, and it will enable a covalent bond to happen between the backbones of the two amino acids that are in a row now, in this case alanine and valine. This being a covalent bond means that the ribosome does have some enzymatic activity. It does allow for covalent bonds to happen that might not otherwise have spontaneously happened. It manipulates the shape of the ends of those two molecules so that a water molecule will pop off and a bond will be formed. What type of interaction is that? Is that hydrolysis or dehydration synthesis? It's dehydration synthesis. You're popping off a water molecule, you're getting rid of a water molecule, you're dehydrating something to build something to synthesize. As the mRNA ticker tape moves through the ribosome continues reading that mRNA in three-letter chunks called codons, bringing in the complementary tRNA molecule that's attached to an amino acid, stapling, covalent bonding by dehydration synthesis, the backbones of two amino acids together, whatever it just brought into the one next to it, and then feeding it on down the line. Those amino acids again form one long sort of pearl necklace style structure called the primary structure of a protein. The primary structure of the protein will spontaneously fold into beta sheets and alpha helices on its own based on the interactions of the tails of the properties of the tails of the amino acids, the R groups of the amino acids. Here's the last video in the slide. Again, this is going to be linked elsewhere because this doesn't play automatically within the slides here. And this is going to show you a video of translation. You'll see the mRNA find its way out of the nucleus through a pore. That mRNA was made inside the nucleus. Transcription happens inside the nucleus. Translation happens outside the nucleus. The mRNA will get clamped down by a ribosome. The ribosome will find start codon. It'll bring in the complementary tRNA attached to amino acid, and it'll shift the whole thing down. It'll do the same thing, read the RNA, bring in the right tRNA, staple the two amino acids that the tRNAs are carrying together, and then shift it on down. And you can watch the protein be formed, that primary sequence of the protein be fed out of the ribosome like ticker tape. I strongly recommend that you watch these videos in order, and you also go back to the macromolecules lecture where I talk about how proteins are made, how the dehydration synthesis happens along the backbone only, how that forms a chain of covalently bonded amino acids, how the R groups are not covalently bonded to one another, but are free to interact to form the primary or to form the secondary structure and the tertiary structure.